A real pleasure to welcome Professor Marina Sirota to this talk series. Marina is an associate professor at the Baker Computational Health Sciences Institute at UCSF. And before that, she was at Stanford as a lead research scientist in systems medicine. And before that, she was in industry working in Pfizer for 3 years, where she worked on developing precision medicine strategies in drug discovery. Her academic education was entirely bachelor's, master's, PhD at Stanford. So she has been in the Bay Area for a very long time. She's happy to take questions during the talk. So as usual, you can put it in the chat or there'll be time for questions afterwards as well. So over to you, Marina.
Excellent. Thank you so much, Suresh and Alex, for inviting me. It's my pleasure to be here. And let me share my screen. Hopefully, you guys can see the regular view, right? Yes. Okay, excellent. So it's my pleasure to be here and tell you a little bit about our work leveraging both molecular and clinical data to better understand Alzheimer's disease. And as I understand, this seminar series is predominantly focused on methodology. We will talk about methodology today a bit, but we will definitely be more focused on applications and translating some of the discoveries that we make using computational approaches. As Suresh mentioned, I'm part of the Baker Computational Health Sciences Institute at UCSF. I came to UCSF 8 years, almost to the day, on April 1st, actually 2015. So I've been there for a little while. This work has been predominantly supported by several grants from the NIA, which I'm very fortunate to have their support for now for a little while as well. So I always start by saying why I'm excited to be a computational biologist or a bioinformaticist today. And that reasoning hasn't really changed in the last 15 years. It's mostly driven by opportunity and by opportunity to leverage and integrate different types of data. On the molecular side, there's tons of data that's available in the public domain. These are just some of the resources, including 1,000 genomes, exact, pretty much any transcriptomics dataset that has ever been published on and beyond is available through the Gene Expression Omnibus. And now that includes more than transcriptomics. It has methylation, single cell data, and so forth. The cancer community has put together an amazing resource called the Cancer Genome Atlas, where they've taken over 10,000 cancer samples and profiled them with multiple omics measurements. And in the context of Alzheimer's, actually, I don't have it on here, I should. The AD portal is quite amazing, AD knowledge portal that's heavily supported by the NIA. There's also drug-related data sets like links that allow us to look at molecular perturbations of different drugs and compounds and leverage that for discovery. And finally, there are clinical trial data sets such as IMPORT. So all of these are quite spectacular. Also, in the last 20 years, or probably even more, people have, or scientists have come up with creative ways to leverage technologies because a lot of these technologies have gotten cheaper and cheaper. So we've been able to profile the genome and the transcriptome now for a long time, but now we can look at antibody repertoire, epigenetics, proteomics, metabolomics. We can look at the microbiome. And many of these measurements we can also make on a single cell level, which provides an amazing opportunity to look at cell-specific signals, but also analytically provide many challenges that are interesting for us to solve computationally. And finally, the last type of data that I'll mention here, and I'll be focusing quite a bit today, is the opportunity to use real-world data. And what I mean by that is electronic medical records. These are data for real patients. It's basically all the information that gets captured when you go to see a doctor, including demographics and some information on social determinants of health. That is definitely an area that needs to be expanded more and more. But we have diagnosis codes, lab test results. We have information on procedures that these patients go through. And by these patients, I mean all of us. So at UCSF, we have an incredible resource. We have data, de-identified data from the UCSF hospital that includes about 5 million patients, including all of that information that's available for research. We also have de-identified clinical notes. My team hasn't explored that to its full capacity yet, the clinical notes piece, but also that provides an amazing opportunity for NLP approaches and other applications. And in addition to that, one of the efforts that Atul Butte, who was actually my graduate school advisor, but also the director of our institute at UCSF now, has been leading for the past 8 years, is to aggregate clinical data across the 6 UC hospitals. And that is, I believe, about 20 million patients. It's a huge proportion of the California population. It's all de-identified and it's available to any researcher at UCSF with no IRB needed. So the whole dump of the data, you can ask any question you want, which is quite incredible. So putting all this together, what my lab is really interested in is figuring out how we can leverage computation to understand health and disease across the lifespan. We tackle a number of different disease areas. We start with pregnancy outcomes, like preterm birth and recurrent pregnancy loss. We study autoimmunity, so diseases like rheumatoid arthritis, lupus, multiple sclerosis. In the past, we've done work in the areas of cancer immunology and organ transplant. I would say those are kind of on the back burner currently, but there's a lot of work in the lab focusing now on aging, neurodegeneration, and Alzheimer's disease. And that will be the focus of my presentation today. And I will say across all of these, you can see a theme. A lot of these conditions, autoimmunity and Alzheimer's, for instance, are more common in women than they are in men. So we have a large focus on studying women's health. And stemming from my also, I would say, graduate school interest is I have a strong interest in immunology. So across all of these projects, we're interested in the role of the immune system. How does the maternal and the fetal immune system interact? Why does the immune system end up turning against self in the context of autoimmunity? And also, actually, when we start looking into sex differences and why Alzheimer's is more common in women than it is in men, we see a number of immune pathways coming up there. So immunology is incredibly complicated, but it creeps into everything. So that's sort of the lens that we're interested in from kind of the molecular side. And the types of questions that we ask are, can we use data integration to be able to diagnose disease better through biomarker discovery? Can we understand disease mechanism a little bit better using data-driven approaches? And finally, can we impact therapeutics? And within therapeutics, we're really interested in, or have been working quite a bit in the area of computational drug repurposing, or finding new uses for existing drugs. So today I will tell you several different stories of ways that we have leveraged both molecular and clinical data to impact drug discovery and understand Alzheimer's disease a little bit better using these resources. But before I get into the first study focusing on drug repurposing, I would like to pause for a second and see if anybody has any questions. All right, so the motivation behind drug repurposing is fairly simple. It takes about 15 years and millions of dollars to take a new drug and bring it to market. And 90% of drugs fail in early development. One potential solution to that is drug repurposing, which means that we can look for other indications of drugs that are already FDA approved. So they've gone through the safety testing, at least for their initial indication, and we can also talk about how that changes from one indication to another, which it can. So as a result, the cost to develop a drug is much cheaper, and it also reduces the risk of failure. There are different approaches for computational drug repurposing, and these are just some of them. There's target-based drug repurposing with the idea that we can leverage genetics. One very famous example of using genetics for drug discovery and repurposing is in the context of cholesterol lowering. There have been some genetic studies by Helen Hobbs and her team at UT Southwestern that have identified PCSK9 as a gene that's very much rare variants in that gene are associated with much, much lower cholesterol. So there's been a number of drugs, antibodies actually developed against PCSK9 that have been incredibly successful at cholesterol lowering. So that's an example of using genetics. There is disease-based drug repurposing, or what I call disease-based drug repurposing. And there the idea is if two diseases present in a similar way, either using their clinical profiles or using molecular data, then maybe a drug for one disease can be repurposed for another. And that has been tried extensively in the context of autoimmune diseases, with some success and some failures. For instance, some of the drugs that are successful for treating rheumatoid arthritis are not necessarily successful at treating multiple sclerosis or something like that. But that approach has been tried. There's molecular modeling approaches and docking-based approaches that look at the structure of compounds and their ligands. And I'll be honest, I'm not an expert in any of that, but there's many smart people working in this area, both in industry and academia, and maybe even the seminar series has tackled some of the examples of using molecular modeling for discovery. And finally, there are network and global signature approaches, where the idea that one drug, one target, one disease might not be sufficient for complex diseases. And this is really the strategy that we have taken. So the question that we wanted to ask was, can we use public data to systematically predict relationships between drugs and diseases? So we wanted to use expression data specifically. And I'll be honest, this is stemming from my own graduate work now a number of years ago at Stanford, but you'll see how we've developed the approaches. So imagine you have expression data from a disease of interest where you have healthy and disease individuals. And you have drug data. And by drug data, I mean cell lines that are treated with a certain compound. So we can compute the expression effect of a therapeutic treatment. And we want to connect the drugs and diseases using gene expression as a proxy. So what we wanted to end up with is a matrix of diseases and drugs where each cell of that matrix is some sort of a score that gives us an idea of how likely a given drug is to be a therapeutic for a certain disease. So what do we do first? We go out and look for data. On the drug side, on the disease side, we started with data from the Gene Expression Omnibus, the public data set that I mentioned. It is incredibly extensive. Right now, it has over 4.5 million samples across many different platforms, but the more impressive number is this 160,000. That's 160,000 different experiments. Many of them deal with disease, many of them are human. We, in our initial study, applied some natural language processing to select out the disease-specific experiments and also those that have cases and controls so that we can define a disease signature. On the drug side, we've been using a resource called the connectivity map. This is a collection of expression data from cultured human cells. They actually use cancer cell lines, and this is a huge caveat, and we can talk about the drawbacks and the benefits of this resource. The original dataset consisted of 164 drugs across 450 experiments, and the updated dataset is about 1,000 drugs across 6,000 experiments. So they look at different doses and different cell lines, mostly focusing on FDA-approved drugs, but also some non-drug bioactive small molecules. This dataset looks at expression genome-wide. It's microarray, so the technology is a bit outdated in terms of what it can capture, but it's genome-wide expression, so across over 20,000 genes. There is another data set I mentioned before called LINKS, which is a follow-up to the connectivity map, where they looked at over 10,000 different drugs, if not more, but they only look at 1,000 landmark genes. And in our hands, with the caveat that these landmark genes can be used to impute 80% of the genome. the transcriptome. In our hands, the imputed data is not very strong. So we've leveraged that data set to some degree, but mostly we focus on using this one. And the hypothesis is very simple. The hypothesis is if a disease signature can be reversed by a certain drug, then maybe that drug could be a therapeutic. We see situations where the signatures are similar, and of course, in many cases, there's no clear relationship. So we developed a computational pipeline to take a disease expression signature, query it against the drug data, and we come up with a drug disease score. The drugs that are similar to the disease have a positive score, and the drugs that are opposite to the disease have a negative score. You can think of this as a correlation. It's actually a rank-based, Kolmogorov-Smirnov-based test that we're using here to identify these compounds. From the initial study that was published, as I mentioned now, a number of years ago in 2011 in Science Translational Medicine, we looked at about 100 diseases and the 164 drugs, and this is a matrix summarizing those results. The therapeutic scores are shown in green, which are the negative, that reverse the situations where the drug might be able to reverse the disease signature, and the positive relationships are shown in red. So for this first application, as I mentioned, the methods paper was published in 2011. The first application was in inflammatory bowel disease and IBD. Since then, we've applied it to some autoimmune diseases like dermatomyositis. Cancer is a natural application because the drugs are tested in cancer cell lines. So we've looked into that as well. We've looked at pregnancy outcomes like preterm birth. COVID-19 was a big one, obviously. During the pandemic, we've applied this approach to COVID-19 to see what we can identify from the FDA-approved drugs. But today, I'd like to focus on our work in Alzheimer's disease specifically. So the story that I will tell you is looking at apoE-specific Alzheimer's disease, apoE4 specific, and identifying therapeutic candidates for that. This is an incredibly close collaboration with Dr. Yidong Huang at the Gladstone Institutes, who's an expert in Alzheimer's disease. And it was led by Ali Tubbs, who was actually my first graduate student at UCSF. The work was published in Nature Aging in 2021. And before diving into the details of this study, I wanted to pause for a second and see if anybody has any questions. All right.
Seems good. Yeah.
Awesome. All right. So here we wanted to take a precision medicine approach to drug repurposing. Specifically, we wanted to look at expression signatures if we stratify patients by their apoE genotype. You might know that apoE is the biggest genetic risk factor for Alzheimer's. So we took the data that we had that we gathered from the AD Knowledge Portal, which is a resource I mentioned before, and stratified the patients into apoE 3.3, 3.4, and 4.4 individuals. We computed differential gene expression for every one of these groups, and this is what the underlying expression data looks like. You can see the distribution of control and AD samples. One big challenge with looking at apoE 4.4 disease is that it's really difficult to find controls. Because it's such a huge risk factor, the individuals that have the apoE 4.4 genotype but don't develop the disease, it's a very small number of individuals that exist that are resilient. So it's always a challenge in these types of studies to explore. You can see the data set colored by case control as well as by apoE genotype. These are dimensionality reduction plots where each dot is a patient. So you can see some separation both by phenotype and by genotype. When we start looking at the differentially expressed genes, you can see that there is some signal. So this is looking at upregulated genes, downregulated genes, as well as the pathways. You can see that there are some that are in common across the genotypes, but a lot of the signal is genotype specific. So in blue, you can see apoE33, in red, apoE44, and in gray, apoE34. So then we took each one of these signatures, as well as the combined signature here on the bottom right quadrant, and we looked at the therapeutic predictions, focusing on apoE44 disease because it's the most severe, and also because Dr. Huang's lab specifically studies apoE44 disease, and they have a great mouse model to explore the therapeutic potential of the drugs in that mouse model. We saw that bumetanide was one of the top hits that reverse the expression of apoE44 disease. So that's here on the bottom of the negative side. It is also has a therapeutic score across 3.4, 3.3, as well as the overall, but it's not close to the best. This is an interesting drug, and we decided to follow that up further. This is just visualizing that expression reversal where the disease signature is on the top and the drug data is on the bottom. You can see that it's not perfect. If we go back, I don't have these slides here, but if we go back to some of our cancer predictions, these reversers are much, much more clear because we're able to match the cell line exactly to the cancer type that we're looking at. For instance, we can look at effects of bumetanide or any other drug in breast cancer cell lines for if we're looking to identify new therapeutics for breast cancer. Here, we are looking at the change at the delta between treated and untreated cell lines, but the reversals that we observe in other diseases are not as strong usually. So this is definitely a drawback. We would love to have the relevant cell types, the brain cell types, treated with these compounds, but that does not exist at the moment. So what is bumetanide or Bumex? It's a loop diuretic. It works by decreasing the reabsorption of sodium by the kidneys, and it's used to treat edema and hypertension. And the first study that we launched was to look at bumetanide treatment and see whether it rescues learning and memory impairments in apoE4 knock-in mice. I'm not a mouse expert, but I will try my best to explain. The mouse model itself is a knock-in of the human apoE4 gene. So the mice that are apoE44 display a memory deficit, while the mice that are apoE33, they do not. So this is the overall, what the overall study looks like. As I understand, mice do not like to be in water. This is a pool that's divided into different quadrants, and one of the quadrants has a hidden platform that allows the animals to escape. So the graph on the right measures escape latency across time. So how long it takes the mice to escape. And you can see in black are apoE33 animals. So their learning rate and escape rate is the same. The solid line is untreated, the dotted line is treated with bumetanide. However, in the apoE44 animals that are untreated, this is the red solid line, there is a significant delay in escape latency across time, which is significantly reduced by bumetanide treatment. This was incredibly interesting and exciting for us to see because we could actually see an effect of this drug on the mouse and its learning ability. We've also followed up with single nucleus sequencing, looking at the effect of treatment with bumetanide across different brain cell types. So this is data from apoE knock, so same mouse model treated with bumetanide versus not. And here, this is a dimensionality reduction plot as well. Each dot is a cell. They're clustered and labeled by different brain cell types, relevant cell types like neurons, different types of neurons, astrocytes, and so forth. And here, their same plot is colored by whether they come from a mouse that was treated with bumetanide versus not. And you can see a clear shift in expression. when we start looking at reversal of the disease signature, so this is that AD signature, the human signature that we were looking at, you can see that a number of different cell types exhibit reversal with bumetanide treatment, and that's shown here as well. We've also done electrophysiology to look at how bumetanide treatment changes the electrical signals in the brain of these mice. I don't have time to get into those details right now, but nonetheless, it allows us to see that the drug has an effect on different levels. But what I wanted to tell you more about in more detail is how we leverage now real-world data, human data, from the electronic medical record to further explore this question. So, as I mentioned, the electronic medical records data at UCSF, at least at the time of this study, had data from 2012 until today. We had about 1.3 million patients, and the data included allergies, diagnoses, codes, encounters, immunizations, and lab tests, medication orders, and well, basically all the information that gets captured when you go to see a doctor. This is what the overall demographics look like. This is patient age across time. You can see that it's a fairly diverse group, roughly equally split between males and females. So we launched this study, and we leveraged the data at UCSF first. As I mentioned, we started with 1.3 million patients, identified those that were on bumetanide, that's 5,500. Some of them did not have complete demographic information, and we wanted to focus on the older population because that's the population that's more likely to develop Alzheimer's. So we restricted to age over 65, which gave us about 1,850 individuals. Then we carry out propensity score matching to identify a case control group. And here we use the 1-to-2 ratio. We matched on age, sex, race, and ethnicity, so demographics, as well as the indication of bumetanide, so hypertension and edema. So we wanted the patients in both the case and the control group to have similar indication, because there is a link between hypertension and Alzheimer's as well. And then we look at the prevalence of Alzheimer's in the two groups. So this is what the UCSF data looks like across 10 iterations of the propensity score matching. You can see that in the bumetanide cohort, there is a significantly lower chance of those patients developing Alzheimer's in comparison to the matched controls. And this is true across the board. Then we repeated the same, at UCSF, this was led by, the analysis was led by Edith Kosti and Tomiko Oskotsky in my lab. We then partnered with a group at Mount Sinai led by Benjamin Glicksberg, and the person who was in charge of the analysis was Masoud Bijak. They started out with 3.9 million patients, so obviously Mount Sinai, very different demographics across the country. About 800,000 had a medical record, about 3,000 exposed to bumetanide, and about 2,400 individuals over the age of 65. We did the same propensity score matching, and the results were incredibly consistent. So you can see that the bumetanide, the exposed cohort, has a significantly lower prevalence of Alzheimer's in comparison to the control group across the board. So this type of analysis doesn't tell us anything about causality or direction, and of course, clinical trials still need to be done, but it's a way to use molecular data to derive a hypothesis. Of course, then animal validation, which takes a long time and is very labor intensive, but we can end it with sort of I would say an in silico clinical trial to explore what the effect of the drug might be in real human data, because these drugs are already FDA approved and are being used for other indications. So what we've done here is we've established an apoE genotype dependent transcriptomic signatures of AD. We applied the drug repurposing approach to identify bumetanide as a candidate for apoE4 specific Alzheimer's disease. We saw that treatment with bumetanide rescued AD-like cognitive and electrophysiological deficits in the animal model. And we leveraged single nucleus sequencing to explore the effects post-treatment, both in mouse and human cells. I didn't have time to get into the human cell analysis, but we've looked at that as well. And finally, we used real-world data to show a lower prevalence of Alzheimer's that's associated with bumetanide treatment. Again, it's merely association. The one other caveat that I will mention is that we actually do not have apoE genotype information on the patients in our electronic medical record. So while we do observe an overall potential therapeutic effect or preventative effect, we're not able to do the genotype stratified analysis like we've done for the discovery. So that's something that we'd love to follow up on, and there are resources across the country and beyond that do have linked genotype and clinical information, but we don't have that at UCSF. And at Mount Sinai, a proportion of the patients have genotype information, but that proportion is fairly small. So we didn't have the power to ask that question. I will pause there for a second. Any questions about any of the analyses, the approaches, the discovery?
I have a few questions, but they can wait till the end, so no issues.
Okay, sounds good. Well, let's move forward then. So what's next? We are currently leveraging single cell data for the discovery aspect of things. So in the past several years, there's been a number of publicly available single cell brain data sets looking at Alzheimer's and controls. We've looked at apoE specific signals. Unfortunately, we did not get enough apoE4 controls. Well, the single cell experiments are incredibly expensive. You get a larger power to discovery because you're looking at a cell at a time, but obviously we're not able to profile hundreds of patients. So the studies that we've looked at is, about 20 cases and 20 controls or something along those lines. So this was a study led by Stella Balanwu in my lab that was published in Frontiers in Aging last year, looking at apoE specific signals. Yechao Li, who is currently a student, has been working on adapting, this is just an example of comparing apoE 3.3 versus 3.4 across different cell types and how those signatures might be different. But Yechao Li is currently a graduate student in the lab who's been taking some of the single cell expression data and adapting the computational drug screening approach that I mentioned to come up with combination therapies. She's using the electronic medical record to screen those data and prioritize them for the animal experiments. And I will say this is very early. This is just a graph showing that we can actually aggregate different single cell data sets across different studies. It's, I will say, no small feat. This is 3 independent studies and just showing that each dot is a cell. On this upper left panel, it's colored by study. You can see that they overlay very beautifully. There are small shifts that you can see globally between case control. That's the graph on the bottom left. And then we can cluster them into specific cell types. So Yechao has been using this data to identify combination therapies that would affect different cell types that are relevant in Alzheimer's. And she has some really exciting animal data that we just got back. Hopefully I'll be able to present it in the near future. That is showing actually a sex-specific effect of a certain combination of drugs that works better in male mice as opposed to female mice, which is really interesting. We have been using electronic medical record data to further characterize Alzheimer's beyond just drug discovery. This is work that's led by Alice Tang, who's an incredibly talented MD PhD student in the lab. The first paper on this was published in Nature Communications last year, and we really wanted to kind of step back and see, well, what does the Alzheimer's population look like at UCSF? we've done some just general visualization and then looking at comorbidities for Alzheimer's and which ones of them might be sex specific, lab test results, as well as the drug data, but more, not in a hypothesis driven way like with bumetanide, but more on a global scale. So this is just showing you some of that analysis. This is looking at the data at UCSF, and for this study, we validated everything at Mount Sinai, again, with our close collaborators from Ben's group. Each dot here is a patient. This is a dimensionality reduction plot based on diagnosis codes, excluding Alzheimer's. So if you look at the patients, and in terms of the numbers, it's the Alzheimer's cohort is almost 9,000 individuals, and the matched control cohort is 2-to-1 matching here again, a little bit over 17,000 individuals. So each dot is a patient, and imagine you take the vector of their diagnosis codes, excluding anything relating to Alzheimer's, and we can see differences. Even though the patients are matched by age and sex and other characteristics, they are inherently different. And we can start looking in both institutions, and we can start looking at what are the comorbidities that are associated with Alzheimer's. Hypertension is one of them, UTI. You can see that the Alzheimer's network of patients is much stronger than the control. So the control, there's very few relationships. And we can look at these in a sex-specific manner as well. What are the differences between males and females with Alzheimer's disease? We can look at specific associations. So this is a plot looking at associations of other diagnoses with Alzheimer's. Of course, we see things like MCI and dementia coming up. But we also see, as I mentioned, UTI, hypertension, hyperlipidemia. And on the other side, we see cancer. So cancer is inversely associated with Alzheimer's. The majority of these associations, about 80% of them, validate in the Mount Sinai cohort. So this is just a log plot of UCSF on the x-axis versus Mount Sinai on the y-axis. We can also look at which of these are sex-specific. Again, there's some that are obviously associated in both, like dementia. There are some that are associated in both with a higher odds ratio in females, like UTI, osteoporosis, hypertension. And then there are some that are sex-specific, which is really interesting as well. And again, the majority of these validated in the independent cohort. We can look at drugs on a more comprehensive scale, which drugs are positively or negatively associated with Alzheimer's. We saw diuretics like furosemide coming up. Obviously, on the positive side, lots of Alzheimer's related treatments like levodopa. So not surprising, but basically gives you an idea of how we can look at this in a global scale. And we can look at this in a sex-specific manner as well. And finally, lab test results. Again, looking at Alzheimer's versus controls, but also trying to understand the differences that we might observe across males and females with disease. So to summarize this study, we've utilized EHRs to look at deep clinical phenotyping and network analysis of AD patients to provide insight into clinical characteristics. In terms of the sex-specific enrichment, we saw that certain associations like osteoporosis, depression, cardiovascular, and musculoskeletal disorders were more common in females with Alzheimer's, and then neurological, behavioral, and sensory disorders were enriched in the male population. We've also, so this first study sort of focused more on sex differences. We've also been exploring the same types of approaches, looking at race and ethnicity and trying to understand whether the disease is different across that demographic axis. This is work by Sarah Walder-Merriam, and it just came out 2 weeks ago in Communications Medicine. The approach is very similar. Now, in addition to UCSF data, we're using the data across the whole UC system. So it gives you an idea of the number of patients that we explore for each. We stratify by different racial and ethnic groups, and then we carry out this type of network analysis. Again, you see huge shifts in disease versus control population, both at UCSF as well as the aggregated UC-wide data, excluding UCSF, obviously. No strong relationships between coloring and visualizing, as well as looking by race and ethnicity. However, when we start looking at the specific associations, this is an upset plot that has the different groups that we looked at, ethnic groups here on the, well, I guess on the side. And then you can see that a number of phenotypes are consistent across all four, but certain ones of them are specific to different racial and ethnic groups, and it's important to recognize. So in summary, we've been looking into some of these questions, integrating molecular as well as clinical data to understand disease heterogeneity and therapeutics. We are investigating specific associations and have been applying predictive modeling to the electronic medical record data as well. So this is the last story that I'd like to tell you about. And this is work also by Alice Tang, who I mentioned before. It's the preprint is out on medRxiv, and the paper is currently under review in Nature Aging. So here, in addition to just general visualizations and association analysis, we really wanted to apply predictive modeling to see whether we can identify patients who are at a higher risk for Alzheimer's disease. But not only did we want to build the machine learning models, we wanted to figure out whether we can link this data to some molecular insights, even though we're starting out with electronic medical records. So for the drug discovery piece, we started with molecular data and then used electronic medical records to try and validate it, or as one level of validation. Here, we're going in the other direction. We're starting with electronic medical records, but we want to get to some molecular insights. And that's this whole interpretation and knowledge network approach that I will mention. So we started out with the current UCSF electronic medical record, about 5,500 patients, about 3,000 of them have Alzheimer's disease. Here we used, you notice that the numbers are a bit different. We used a much more stringent phenotype information. We integrated our EHR data with information from the Memory and Aging Center. So not just the ICD codes, but actual data from an independent matched resource. And we had a number of restrictions because we wanted to look at this longitudinally. We needed patients who were in the system for at least 7 years. So that reduced our sample size quite a bit. So we had 749 AD patients, and of course, a huge number of controls. This is what the results look like. Obviously, we're much better at predicting Alzheimer's 1 day before diagnosis than 7 years before diagnosis, but there is predictive value across the board, as you can see, using both just clinical features as well as demographics. We observed that matching the control cohort by age and ethnicity allows us even better prediction. So this is looking at various fee code categories. So these are the categories of the clinical features and their association across time. This is looking into some of those details, diving into the actual fee codes themselves. Some interesting associations that we observe include hyperlipidemia, osteoporosis, dizziness, vitamin D deficiency, atrial fibrillation. So many of the associations that we observed previously, here we can say are actually predictive of somebody being diagnosed with Alzheimer's, even 7 years out. we can also do this analysis in a sex stratified manner. So we can see that there are some features that are female enriched, including osteoporosis, myocardial infarction, and others. And there's some features that are male enriched in terms of their associations. And of course, some things that are associated across the board, like hyperlipidemia. And this is diving into some of those associations as well. So then the next step, as I mentioned, was to overlay some knowledge networks to try and get to the interpretation of these clinical findings. So SPOKE is a data set, a knowledge network that has been developed by Sergio Baranzini and his lab at UCSF for a number of years. It aggregates a number of different, I think over 40 different knowledge bases, and has different types of concepts like genes, drugs, diseases, and so forth. So here you can see the clinical features on the right-hand side, and then tying to Alzheimer's disease on the left-hand side with molecular features in the middle. And you can see these little pie charts. They demonstrate which of the features are consistent across the different time models that we presented. So let's dig into an example. If we look at the clinical features that are consistent across all the time models, we have familial hyperlipidemia and osteoporosis. If we go back to the molecular features that we extracted using SPOKE, these are the features that we end up getting. So we wanted to investigate and validate some of these associations. The first one we looked at is between hyperlipidemia and osteoporosis. We leveraged the data from the whole UC system, doing an analysis using the Cox proportional hazard ratio. And we saw that the link between hyperlipidemia and Alzheimer's was consistent across this independent data set. We also saw that it was consistent in both males and females. When we look at osteoporosis and Alzheimer's risk, we also see that it's associated in the independent population, but we see that the association is significant in women, but it's not significant in men. So then we wanted to bring in genetics into this picture. So we partnered with Gabriel Serrano from Sergio's lab to carry out co-localization analysis, which basically means we can take GWAS data from both phenotypes of interest and see if there's anything in common, any common genetic signals that co-localize to the same locus. I'm not going to get into the details in the interest of time, but this is what the data looks like for AD and LDL, which is the cholesterol phenotype. Using this analysis, and this is a Manhattan plot where you see the log p-value on the y-axis and different phenotypes on the x-axis, that the specific region of the genome that's responsible for apoE, and again, we know this is the biggest risk factor for Alzheimer's, has a significant peak around hyperlipidemia and Alzheimer's disease. This is not, we see a similar co-localization on the protein level as well. This is not incredibly novel. We know that this link exists, but it's quite remarkable that we can get to a hypothesis from the clinical data and observe that association in completely independent molecular data set. So looking at the link between hyperlipidemia and Alzheimer's, we did see from SPOKE that apoE is very important and it's coming up. But then we wanted to investigate the other link between Alzheimer's and osteoporosis. So we gathered two independent data sets, an Alzheimer's data set, as well as a heel bone mineral density, GWAS, and that's a measure for bone health. And this is what the data looks like. This is plotting p-values of the heel bone mineral density study against the Alzheimer's study, stratified by females and males. And you can see that there is a peak, a common peak in females that we don't observe in males. This is around the MS4A6A gene. We can also look at the fact that this association is using it EQTLs, so now bringing in expression as well, so some functional data, that this relationship is female specific. So this is looking at Alzheimer's overall. Unfortunately, we did not have a stratified data set for Alzheimer's, but we did have a stratified male and female data set for heel bone mineral density. And you can see that the peak exists in females, but not in males, when we look at the expression data tied to that locus. So this is far from causality and mechanism, but to us, it was really interesting to see that we can now not only identify known associations, but also discover new genetic associations using this approach, starting out with clinical data and then layering on knowledge networks and genetics on top of that. Looking at the network itself, so that specific gene did not come up in the knowledge network. There's a number of other immune genes that did when we explore the link between Alzheimer's and osteoporosis, like IL-6, TNF, and others. So we do believe that there's some immune component to this relationship, but that's something that needs to be explored much, much further. So in terms of our other relevant work in this space, we're working on applying clustering approaches to identify Alzheimer's subtypes using HR. We're very interested in looking at characterizing AD and other related dementia heterogeneity with EMR data. We had a summer student in Vienna who just gave a talk at the AMA Informatics meeting about that work. We're interested in understanding the impact of social determinants of health. That's led by Silvia Miramontes, who's a graduate student in the lab, and also explore some of these hypotheses. So for instance, we're looking more in detail into autoimmune diseases and relationships between autoimmunity and Alzheimer's, as well as cardiovascular disease factors. With that, I hope I gave you 2 examples of ways that we're leveraging both clinical and molecular data in the drug discovery space. We start with molecular and move into the clinical. With the predictive modeling, we start with the clinical and move into the molecular. And these are approaches to integrate clinical and molecular data when you don't necessarily have them on the same patients. And I'm hoping that can bring us a little bit closer to precision medicine for Alzheimer's disease. With that, I'd like to thank everybody on my team who's been leading all of this work. The drug discovery work was led by Ali, but also in close collaboration with Tomiko and Edith, who did the EHR piece of it. Alice has led a lot of the EHR data analysis, and Yachao is moving forward with some of the combination therapy prediction. Sarah led the study on the race and ethnicity differences, and we're moving in all of these directions that I mentioned. Also, many of our collaborators, Yudong Huang and his team at the Gladstone Institute, as well as Ben Glicksberg and his team at Mount Sinai, and many, many others, as well as our funding sources. This was us in February of 2020. Now we're managing this hybrid life, but we do get to see each other and hang out. And with that, I will stop there and I welcome any questions.
Cool, thank you. It was a great talk. Are there any questions? I have several, but I'll take audience questions first. Anybody? Any in the chat? Okay, I guess I'll ask one first. It's very naive and very general question, right? Okay, we have a question from Benjamin Rutsky, who wants to know whether you corrected for the difference in osteoporosis prevalence in males and females.
Yes. So, well, I guess, what do you mean by corrected? Our cohort is matched by sex for the overall predictive model. And then we split into males and females and do the analysis separately for males and females in the EHR to see whether that feature comes up as predictive or associated. So that's the strategy that we have taken. Yes, osteoporosis is less common in males, but so is Alzheimer's disease. So we don't correct for the indication of the diagnosis of osteoporosis, but we do match by demographics, the cohorts that we look at.
There's another question that wants to know how the data integration was done across the different snRNA-seq datasets in Alzheimer's.
Yeah, we use canonical CCA here to aggregate the data. There's a number of different approaches to data aggregation. We've also explored Harmony as an approach, an existing algorithm to aggregate single cell data, and we've been fairly successful with both. This was just 3 studies. We're also have some ongoing projects in the autoimmune disease space to integrate across many more data sets. So it's definitely of interest to the group.
Cool.
Sorry, I'll mention one more thing. The bigger challenge is some of the single cell, there's also single nucleus data that we haven't done. We haven't integrated single cell and single nucleus data together because of just the technical differences in these data sets.
Cool. Yeah, I guess I'll sneak in one of my questions, which was, so I was generally very curious because there's all these factors, right? It's very, very, very mixed. You have many, many potential factors acting linearly, non-linearly. So in particular, I was interested in this connection between the disease correlations and the drug correlations. So to what extent is a specific drug, right? So you have bumetanide, but the neighboring drugs that are all used for treatment of the same kind of edema, right, renal dysfunction studies. So can you disambiguate the sort of the drug disease correlations that you were talking about in the second half with the drug correlations that you were talking about in the first half? If you know what I mean, does it actually, can you, go ahead, yeah.
I think I understand. So on the molecular side, it's based on which genes they perturb. And we have seen, there's a different study that we did just clustering the drugs based on their expression profiles. And we saw that some of the drugs that are used for the same purpose clustered together, but some of them have drastically different expression profiles. So they act in different ways. Some of the drugs are just really dirty. So they just affect expression of everything under the sun. And for instance, like HDAC inhibitors come up a lot in many of our predictions just because they perturb so many genes. Or metformin is used for a million different indications from diabetes, it's an original indication, but also cancer and PCOS and other things. So it comes up a lot as well because it just affects so many things. So on the expression side, I would say it just really depends. It's incredibly heterogeneous. And that's why we need follow-up studies to understand the mechanism of action. I don't think we're going to get that from expression data alone. We might be able to say transcriptionally, these drugs are, and sometimes you have drugs that act on very different diseases that cluster together. So we've seen it all. It's, I wouldn't say random, but it's incredibly heterogeneous. On the EHR side, with respect to bumetanide and other diuretics, that's actually, it's not something we explored in this initial study, but that's something that we're actively looking at right now is what is the relationship between Alzheimer's and other diuretics? Can we understand that? Can we better understand what is it about that drug? And not only in the Alzheimer's indication. So for instance, for pregnancy outcomes for preterm birth, we identified lansoprazole, which is an antacid, and it's incredibly safe. We are having a hard time exploring it in the EHR because it's over the counter. So that's not really captured, but based on expression, it looked really nice. We see some effects in an animal model, well, animal models of pregnancy. I mean, that's a whole different hour-long discussion. They're not great, but we want to look at other antacids. Is it the antacid or is it the mechanism of action of that drug, that specific drug, lansoprazole, that might be effective? But I would say for both, we need follow-up studies. So in the EHR, we can sometimes look at it. So with Bumex, we can, and we are looking into that right now. But also, additional studies to understand the mechanisms are really
Yeah.
needed.
I was curious about one thing. If you applied this
Yeah.
entire strategy to historical data, how well, where prospective trials have been conducted,
This
how
is a
does
great
it work?
question. We haven't done that. We should. The problem is the clinical trial data. I mean, there's data in clinicaltrials.gov, but overall it's incredibly messy and it requires hand curation. So that's why we haven't done that across the board. I think there are some studies out there that have looked at drug repurposing in kind of a global approach, but we haven't done that. We've focused on specific diseases where we had a collaborator who we can work with to validate them, not kind of a global view for the majority of our studies.
Any other questions? I was curious with bone mineral density, apoE4 is also linked with that, right? But it didn't show up in your, so it is, I just Googled it to find out that apoE4 is linked to higher osteoporosis risk, right?
Oh, interesting. So we did not, so apoE we found was in the context of hyperlipidemia. We didn't see it in the context of osteoporosis, but that's super interesting. We should explore that as well.
Because I just Googled it, the risk of osteoporosis in apoE4 versus apoE3 to see if there's a link. It seems there is. That's the first result that showed up.
Excellent. Well, we'll take a look at that.
Great. If there are no other questions, then thank you very much for this fascinating talk. And yeah, that was great. Thank you very much.
Thank you. It was my pleasure. Thanks for having me.
Bye, everyone.